everybody. I am the Patient Engagement Manager for the EURO-NMD Patient Advisory Board, and I'll be moderating this first EURO-NMD patient webinar series. So the first one is about gene therapy. First of all, I would like to tell you that don't hesitate to ask questions on the chat. There'll be a Q&A at the end. So we have two presenters. First of all, Olga Germanenko. She is a patient advocate from Russia. She's the founder and current president of the Russian SMA Family Foundation and vice president of SMA Europe. Her relationship with neuromuscular disorders is personal. She's the mother of a 15-year-old daughter living with spinal muscular atrophy type 1. And then we have Professor Annemieke Aartsma-Rus. She's a professor of translational genetics at the Department of Human Genetics of the Leiden University Medical Center. She played an important role in the development of antisense-mediated exon skipping for the Duchenne muscular dystrophy during her PhD research. Her work currently focuses on developing antisense-mediated exon skipping as a therapy for Duchenne muscular dystrophy and rare brain diseases.
You want me to start the presentation?
Please do.
Yes.
Yeah.
Okay. Sorry.
I
will. Okay. Wait until I do some technical things to make sure we share on the right screen. There we go. Oh, no. There we go. Okay. So I'm going to assume that if you don't see my screen in full slide mode, that you will tell me, and I will get my pointer. So thank you very much for attending this lecture. Annemieke and Olga have tried to to devise the labor. So my focus will be on Duchenne. Olga's focus will be more on spinal muscular atrophy because that's the diseases that we have most experiences with. But you will realize that many of the challenges and the knowns and the unknowns are similar between the diseases. So this is not really relevant for this talk because it has to do with my my exon skipping work. But just for full disclosure, I give all my disclosures, and well, feel free to take a screenshot if you want to read them later. Sorry. So today we will talk about gene therapy, and I will try to explain the basic principles because it's good to understand the basic principle, because then you will also understand the challenges and the risks and what we know and what we cannot know and what to expect. As I said already, the focus of my talk will be on Duchenne, but I'll mention SMA sometimes as well. What is the current state of the art for Duchenne, and also what are the long-term expectations and the things that we don't know yet, because bear in mind, gene therapy is still very much in a pioneering stage. So first, something about chromosomes and genes, because gene therapy, the word gene is in it. Genes are the codes for proteins. So our body, each of our cells has the code of all the different proteins that we need to make, and these genes are located on the chromosomes. So each of our cells has 2 meters of DNA in these chromosomes, and they come in pairs. So each cell has the same pairs, 22 pairs are the same in males and females, and then males have an X and a Y as the 23rd pair, and females have 2 X's. And on these chromosomes are the genes, as I mentioned already, and the genes contain the code for the protein. So our body translates the gene code into the protein code. And if that happens, you can understand that if there's a mutation on one of the chromosomes in one of the genes, that means also that there is a mistake in a protein. So there will be mutations in a gene that prevent the protein from being formed. And of course, if that happens, that can cause a disease. So in case of Duchenne, there's a mutation in the gene for dystrophin, so no functional dystrophin can be produced. And what does dystrophin do? It connects the skeleton of the muscle fiber to the connective tissue surrounding the muscle fiber. And I like to use the explanation of a carabiner because it has two important domains that you click with, and it forms a connection. If you don't have dystrophin, there's no shock absorbing function, so muscles are not stabilized during contraction, and then you lose muscle tissue because there's continuous muscle damage, leading to inflammation, formation of scar tissue, impaired regeneration, and loss of muscle tissue. And Duchenne patients cannot make a functional dystrophin, as you can see here, and there's an alternative disease called Becker muscular dystrophy, where patients make partially functional dystrophin. So you can see this here, this elastic domain is shorter. And why do I like this explanation of the carabiner, the rock climbing carabiner? It has these two domains, just like dystrophin, but also you don't need dystrophin to have the muscle. You don't need the carabiner if you go rock climbing. You need it when things go wrong, just like you need dystrophin when things go wrong. So if there's no dystrophin, you have a carabiner like this. If things go wrong, things go really wrong. And if you have a partially functional dystrophin, when you fall, I mean, this will be a less comfortable fall than if you have a proper working carabiner, but you can imagine that this is much better than having no connection at all. So that is the cause of the disease. And then, of course, you can reason, well, if there's pathology because the dystrophin gene is mutated and cannot produce the dystrophin protein, why not give patients a copy of the dystrophin gene, and then they can make the dystrophin protein with that. And this is the gene addition therapy, and it's also called gene replacement. But I think gene replacement is a bit of a misnomer, because for dystrophin, what is happening, so we have a mutation here on the X chromosome. What gene replacement would suggest that we cut this out and we replace it with a functional copy. That is not what happens with gene therapy. What happens is that you give an extra gene. So the mutated gene is still there, but you give an extra gene that is functional. And this is called a transgene. Now, of course, it's relatively easy to do that in one cell. So give this functional gene to a single cell, but that's not going to help anyone. You need to deliver this to the majority of cells where things go wrong. So in Duchenne, this is the muscle. In spinal muscular atrophy, this is the motor neuron. So you need to get it to the majority of cells to have an effect. And like I said, this is a separate piece of DNA. So you add something and it doesn't go into the chromosomes. It just floats around in the cell. And that's another thing to remember. Now, some challenges for muscle diseases. Muscle is very abundant, so 30 to 40% of our body weight is muscle. It's not a single organ, so we have about 700 different muscles, all affected in Duchenne. And once the muscle tissue is replaced by fat and fibrotic tissue, this is irreversible, and the loss of function cannot be recovered. So the time of intervention matters. So if we look at spinal muscular atrophy, motor neurons are much less abundant, and it's possible to treat them. It's relatively easier compared to skeletal muscle, but the principle that the time of intervention matters, that also applies to spinal muscular atrophy. Once the motor neurons are gone, you cannot bring them back. So how do we deliver these genes to the different cells? So muscle in case of Duchenne, motor neurons in case of spinal muscular atrophy. We use a viral vector. And what we use for both is adeno-associated virus. And well, this can deliver to skeletal muscle, but it's important because adeno-associated virus is a virus that also occurs, say, in the wild. So it's possible that you have encountered it. And if you have, then you have immunity to it, and then you cannot get the gene therapy. It's a very small virus, which is not a problem for SMA because the SMN gene is small, but it is a problem for dystrophin because the dystrophin gene is huge. So we need to engineer, we need to make microdystrophin. So what are the components that go into a viral vector? Some viral sequences, because your DNA needs to go into the viral particle, so it needs to be recognized. There needs to be a promoter, so it's on in the right tissue. So for spinal muscular atrophy, motor neurons for Duchenne muscles. And in case of Duchenne, the dystrophin code, which here is a microdystrophin because otherwise it will not fit. So we need to make the dystrophin protein code shorter. So this is the full length, and here we have a very short microdystrophin. And if you then look, so this ABD, that would be the first clicker domain binding to actin, the skeleton of muscle fibers. This CIS is the second clicker domain binding to the connective tissue surrounding the muscle fiber. And then this elastic domain normally has 24 repeats and 4 hinges. And you can see the microdystrophin, it's a lot less. So you have 2 or 3 hinges and you have 4 or 5 of these repeat domains. If you ask each of the different companies which one works best, they're all going to say their microdystrophin works best. But if you look at them, you can see there's more similarities than differences. But it's good to bear in mind, this is a microdystrophin. This is much smaller than we see in Becker patients. So it's not going to be fully functional. It's hopefully partially functional. So AAV can deliver to the central nervous system, so the motor neurons. It can deliver to skeletal muscle, and you see that there's different flavors of AAV. Just like with COVID, we had the Delta, with the Omicron. Well, AAV has the same thing, but then we use numbers. And you see that, for instance, 7, 8, and 9, they go to muscle quite well. But what you also see is that they go very much to liver as well. And this is the function of the liver to, well, filter out the things that don't normally belong in the body. So it's not strange, but it's good to bear in mind that even when people say AAV goes to muscle, AAV goes to motor neuron, yes, it will, but it will definitely also go to the liver. So I mentioned AAV is not pathogenic during a normal infection. So probably about 30 to 50% of us have seen AAV. We've had an encounter with AAV. We didn't get sick, but our immune system responded to it. So the fact that it doesn't make you sick when you get a few viruses doesn't mean that you will not get sick when we use gene therapy, because the dose that we use here is much, 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 much higher. And to give you an idea, the amount of viruses that you give to a Duchenne patient for gene therapy is 2,000 times the number of erythrocytes that we have in our body. So the dose is massive. So even though normally you don't get sick, of course, if you use a very, very high dose, you will get sick. There will be side effects, especially since this goes to muscle, but also it goes to liver a lot. And per cell, there will be a lot of viruses in the liver, and that will cause a side effect. Also, the immune system will have an immune response because this is just such an amount of viruses. So even though patients get immune suppression, there will still be an immune response. And this also means that if you've seen an AAV in the past that is used for gene therapy, you cannot get the gene therapy treatment. So if you just had a natural encounter with AAV, your immune system has seen it before, you cannot then give the gene therapy, because this would be a dangerous situation, because your immune system will react massively to the huge amount of viruses. So this is why an antibody test is done during screening to filter out patients who have already seen the AAV in the past, because that would be dangerous. So they are excluded from gene therapy. And this is what I just said. So there's two things why you don't want this. This is very dangerous, but also if you've seen the virus in the past, your immune system will counteract the AAV and it will be less effective. I'm more worried about the dangerous side effects of an immune response than the fact that the efficiency will be lower. But yeah, I think this is just something to bear in mind. And also because there is such a high immune response, patients who don't have AAV antibodies and who will then be treated with AAV gene therapy, they get a very high dose of steroids just to try and reduce the immune response. And even then, there's still an immune response. So where are we at? I'm sure that you are aware that Zolgensma is approved for spinal muscular atrophy. So this is the original name. I will not try to pronounce these or the original names. For Duchenne, Elevidys, so this one it is approved. But there's also, as you can see, multiple in clinical trials or waiting to be tested in clinical trials for Duchenne. Also for limb-girdle muscular dystrophy, for several of the limb-girdles, there are gene therapy trials ongoing, but these are very small in small numbers of patients. So going back to Duchenne, where are we at? I mentioned already Elevidys, the gene therapy, microdystrophin gene therapy from Sarepta, is approved in the US and in several countries in the Middle East. It's approved for ambulatory patients 4 years and older, and it's only approved based on dystrophin restoration. The confirmatory study did not show that this slowed down disease progression, and we'll get back to that. It's also approved for non-ambulatory patients, and their additional trials are ongoing to show functional effects. And it's not approved for patients with deletions of exon 8 and 9, and we'll go into that as well. But say going back to the overall thing, what do we know? There's an approved treatment in the US, and there's clinical trials for multiple companies with slightly different AAV microdystrophins. So we have findings from over 600 Duchenne patients now. And what we know is that gene therapy can allow production of microdystrophin in the skeletal muscle. So in that way, it works. But what we also know is that this is accompanied by side effects, and these can be severe to very severe. Sadly, so far, 3 patients have died after gene therapy. 1 was young, 2 were older, and they all got AAV9. So I think that is important to bear in mind. The patients who died received AAV9. The one that is approved is 7.4. So just good to remember. Side effects, the risk of very severe or severe side effects is related to the dose, but we cannot really go much lower because then there won't be enough microdystrophin produced. It's also related to age, and again, that's indirectly related to dose because these viruses are given on a per kilogram basis. So if you're older, usually you're heavier, and if you're heavier, then you need more virus. So larger dose, higher risk. But also older patients have a more mature immune system, so just more chance that something will go wrong. So these are the acute side effects. So they happen within the first 2-3 months after treatment and primarily immediately after treatment. So nausea, liver damage, kidney failure seen with AAV9, sepsis, and death. So this is not trivial. And I think this is good to remember. There is a risk of a severe side effect. In Duchenne, there's also side effects seen in some patients that occur later, so after 3 months of treatment. And this is breakdown of muscle, inflammation of the heart, and this is due to immune response to the microdystrophin. But it's only seen in some patients. And why is that? Because we can say, well, the immune system will recognize foreign proteins. If I say Duchenne patients don't make dystrophin, and now we give microdystrophin, that should trigger the immune system in all patients. But that's not what's happening. Now, why is that not happening? Because here we have the normal dystrophin, and here we have the microdystrophin. So this is the normal dystrophin as it is produced in muscle. However, in brain, we have a very small dystrophin containing only this part, so the second clicker domain. Most patients, 99+% of patients, have this smaller protein. So this part of the microdystrophin is known for these patients. Now, most patients have a mutation in the middle of the gene, somewhere here. So what they will do, they will make this first part. It's not functional, it's not stable, but the immune system will be familiar with this first part. So again, this part of the microdystrophin for most patients is also known. However, there's an exception. There's about 1 in 10 patients who have a deletion at the beginning of the protein. So they miss this part of the dystrophin. So they cannot produce this. So now we give a microdystrophin that suddenly has this piece that they've never seen before because they cannot produce it. And that will then trigger an immune response, especially the part of the protein that's encoded by exons 8 and 9. And this is why these patients are now excluded from getting the gene therapy, because this autoimmune response to the microdystrophin that leads to muscle breakdown, inflammation of the heart, is very dangerous. And actually, you end up getting worse because all the cells making the microdystrophin are removed. So you end up with less muscle than you had. And of course, inflammation of the heart is also not a good thing. So that were the side effects. And like I said, they are not trivial and there's risks. In part, it's predictable risks like the autoimmune response. In part, we don't really know why some patients get very severe side effects and others do not. What is clear is that patients make microdystrophin after the clinical trials. What we know from a functional effect is currently not that much. So what first was done was open label study. So patients were treated and then compared to the natural history. And then what was seen is that patients did better than you would expect from natural history. So people got cautiously optimistic. But then there were placebo-controlled trials, and these so far has not shown that much effect. So what we know from a first study testing Elevidys, there was no difference in function between treated and untreated patients. However, the treated patients got a suboptimal dose. So maybe that is why. The second trial also did not meet its primary endpoint. So the North Star Ambulatory Assessment Scale, which measures ambulatory function, 1 year after treatment, the placebo group and the treated group did not have significantly differ. So that was Elevidys. Then Pfizer did also a study, and also they, after 1 year of treatment, they did not see a difference between treated and untreated patients. So does that mean that this does not work? Well, personally, I think we don't know. And why do I say we don't know? That's because I think in hindsight, these trials were not very well designed, because they were designed with the idea that microdystrophin will improve the function in treated patients. Well, actually, it's not a normal dystrophin. So what you expect is a slower disease progression. It's really difficult to show a slower disease progression in 1 year, especially in stable patients. And the cohort that was included in these two trials were stable. And of course, if you expect that patients will improve, you will be able to measure that in a stable group. But based on the working mechanism of microdystrophin, that was not really realistic. So now I think in hindsight, we can easily say, well, it would have been nicer to have a longer trial or to have a trial in older patients or both. But well, that is too late now. But it's good to bear in mind, functionally, there is no evidence yet that microdystrophin treatment can slow down disease progression. For Elevidys, sometimes there were secondary endpoints that did improve. For the Pfizer, this was not the case. So the treated and untreated group were almost identical. So there's some more considerations. And the first one is that not all patients will respond equally well. And this is the case for Duchenne. This is the case for spinal muscular atrophy. The patients that you will see on social media, those are the extremely good responders. So that is something to remember, that, I mean, it would be great if your children or your family members respond as well. But what you see on social media, those are the extremes. Those are the best case scenarios. The majority of patients respond less well. In publications, we also see that some patients are declining or responding less well. There's very severe side effects seen in Duchenne, and I think that is important to remember. Usually shortly after treatment, and I think there's especially a worry for older patients because they will need a higher dose, but also older Duchenne patients will have respiratory problems, they will have cardiac problems, so they have less buffer to cope with these side effects. So I think this is something to bear in mind when considering the gene therapy. Then some challenges towards the future, what we don't know. We don't know how functional the microdystrophins will be in humans. We know they're functional in mice and in dogs, but these animals walk on 4 legs, we walk on 2 legs, the dynamics are different. So we need more placebo-controlled data to really see anything, because, well, so far we just don't know. One thing also that we don't know is how long will this gene expression, long will the microdystrophin expression persist? It's a microdystrophin, it's not fully functional, there will still be muscle turnover. So with time, there will be less and less and less cells that make the microdystrophin. And then the question is, when do you stop having enough microdystrophin to have an effect? Is that in 2 years or is that in 20 years? We don't know yet. For SMA patients, they will grow and that will dilute the effect. So also there we don't know, will there be a reduction of effect longer term? We just don't know this. And again, we're pioneering this, so there's just things that we don't know. Then, well, we know that there is an immune response to the AAV, so some patients cannot get it because they have pre-existing immunity. And for a few patients, we have this response to microdystrophin, but this is something that we can so far predict, so we can exclude these patients. So that was my last slide, so I will stop sharing, and then I'm not sure whether we immediately go into Olga's presentations. I think so, and then do questions afterwards. I think, yeah, I'm not.
Maybe we should do questions afterwards.
Afterwards, okay. So then
like
I hand over to Olga for the patient perspective. Thank you.
Thank you very much. Let me also share my slides. It should work by now, I guess. Yes. So if the slides is not working, just let me know. I've been already introduced, so I will not repeat that. So a few words about SMA Euro, because what I was talking about is actually the experience we collected as an organization as well. And I will not basically talk about the medical things, because I think that Annemieke quite well described the challenges and current state of art in gene therapies. And even if she mostly was talking about Duchenne gene therapy, but the same we can really transfer to SMA, because really it's also AAV-based product. And I would most likely talk about the experience we already had with the gene therapy product over this last 5 years. The product is on the market. I would like to start with a definition of gene therapy, because it's really sometimes is unclear what is really gene therapy. But as you can hear from Annemieke, also when we call gene therapy, we mainly address AAV-based products by now, at least. And I think that despite of in SMA having this gene therapy product for more than 5 years, we still see that it's not clear understanding of what is gene therapy for patients. Sometimes it's caused some additional fears because there is a lot of incorrect information around. Also, we do have a lot of advertising which grows unrealistic hopes in the patient communities. But I think that these problems also can occur in some doctor population, because not all the doctors really know well what gene therapy can offer and what could be the consequences of using it. Therefore, there is also the question of educating the doctors. And I think that for general population sometimes it's also challenging to understand what is a gene therapy, because as Annemieke starts with that, sometimes we think that gene therapy is just gene replacement of some problem in our DNA, which is not true. I think that what was already mentioned, that gene therapy is irreversible treatment. So once it's inside of you, it cannot be taken out. So as any other long-term treatments. And I think that it's and it should influence actually clinical decisions of whether to treat with gene therapy or not, but also regulatory and sometimes reimbursement decisions as well. And I think that it's important to note always and have it in mind that gene therapy currently is not a cure, and it's not a cure for all patients with the same disease, and it's not a cure, unfortunately, for some particular ones. I would talk mostly about anasinogene, which is Zolgensma gene therapy, the only gene therapy currently approved for SMA. And as you can see, I specifically put in my label, which is limited. The population can pretend to receive in gene therapy for particular very small number of patients with SMA. So currently it's the patients up to 3 SMN2 copies. And the dosage limit will be about 21 kg. So we having very small number of very young patients who can pretend for gene therapy. But for me, it's more important to give a perspective of how we perceive gene therapy and what we have to communicate and discuss with our doctors when we discuss in gene therapy. I think that the first, we have to understand that gene therapy really not changing anything in our DNA. So as Annemieke already showed very proficiently, that it's really additional component which is going directly to the cells with this capsid, AAV capsid, which is bringing the element into our cells. So it's addition to what is wrong with our DNA, but not changing the DNA as well. I think that important to discuss with the doctor what we can expect from this type of treatment with gene therapy. We cannot expect that our disease will just go on. I know the cases from some patients who even tried to get additional DNA testing after receiving gene therapy just to be sure whether there is still deletion in SMA1 or maybe it was cured with gene therapy. So of course the answer was no, deletion is still there. So we know for sure that it's not fully cured for their disease cause. There is, of course, some limitation in possible effectiveness, which is mostly dependent on how much motor neurons is still exist in the, and saved in the patient. And also I think we always as the patients also have to have it in mind when we talk about what we can expect from the treatment, the current state of the patient, and also how early it is possible to start treatment with gene therapy as with any other disease modifying therapy we have. Long-term effect, we already discussed that, but that is also, I think, to have in mind for how long we can expect effect. We don't know yet. Real term, 5 years, 20, 12 years, 50 years, we don't know, maybe a few years only. Safety is really important things to be discussed, because really there is some specific regime patients should follow and the family should follow before gene therapy. I will talk about this later, during the gene therapy administration, and also after that. The safety the most important part to consider, I guess. And here we really have to have in mind that if we using gene therapy as a product, so we have to understand that we have a short follow-up and long-term follow-up. For the short follow-up, I mean, there are a few months after gene therapy when you have to be under the doctor seeing you very often to care about their immune response and possible side effects you will experience. But also long-term follow-up is important, because sometimes the side effects could be with you for like a year or even a little bit longer. That's what we see in the patients now. If we talk about their response to treatment, because it's currently also the hot topic in SMA, we still do not have an answer what to consider good response to gene therapy and what is suboptimal response, which will require probably to use another DMT after gene therapy. So that is really question without answer by now. But I think that if we're talking about the gene therapy, we have to understand that even if we have a great product, because gene therapy is really working in SMA, and effectiveness of gene therapy for SMA is really proven. And we see a lot of the patients who really have a great result undergoing gene therapy for now. But also, I think it's important to keep in mind that this type of treatment also not solving the full loss of motor function, and we cannot restore what was already what we already lose. And of course, I think that sometimes we expect too much from the gene therapy just because of the name of gene therapy, and also because sometimes the price, because it also affects our impression, and we really expect really I don't know, miracle to say result from gene therapy. And I think that sometimes also we experience wording issue when companies trying to market SMA gene therapy. I just put few words like gene replacement therapy and one and done. This is two combinations of words was used by the manufacturer actually to describe to the patient community and to the market of SMA gene therapy, and it was all both of them was mistakenly perceived by the patient community. So with gene replacement therapy, of course, what we as a patient understand that it will be some changes in our DNA, which I already said it's not true. And with one and done, we sometimes think that one and done means I only need gene therapy and nothing more. So yeah, as soon as I receive gene therapy, I'm good enough. I don't need anything else, which is again not really true. Yes, it's a single infusion. We cannot repeat gene therapy for the reasons Annemieke described earlier. But also, I think that sometimes it's bring the question to think that also follow-up and compliance to the standards of care is not that much important. So we lose this patient compliance for some times. And I think also the great expectation of the gene therapy sometimes bring us to the dissatisfaction with the results because we expect a miracle that disease will go completely, which is not happening in most of the cases. And if the child after gene therapy is not completely healthy, but doing still great, like standing or walking or sitting, depends on them on the initial stage of the disease progression. Sometimes it's bring us also to the raising additional requests for using other disease-modifying therapies after Zolgensma. It's really one-time treatment, but we really, for now, don't know its lifelong effect. And we don't know whether it's really lifelong or for how much time the effect will still with us. And according to the published data, for now we have the data up to 7 and 5 years post-dosing in SMA patients. So we can be sure that in this period of time, patients what patients can achieve, then maintain in terms of the motor function at least. And I want to also raise the question of the dual therapies, because sometimes it seems to be that our doctors think that if gene therapy were there, so patients will not really need additional things, additional DMTs, but that is really what is in discussions. And also the company manufacturer of gene therapy, in their press releases from the early trials, also shows that most of the patients, more than 60% currently on the additional add-on therapy nowadays. But we don't know really whether additional implementation of other disease-modifying treatments will add additional benefit. So hot and hard topic, and current European expert consensus in SMA saying that it should not yet to be used in general practice, this combination and dual treatment usage, but it's already there, and we now having additional clinical trials by the companies to try to see whether there is additional benefits from adding on DMTs after gene therapies as well. But that's still what we don't know. Some problems we have in understanding of the patients, what really can affect them. Again, underestimation of steroids regimen and possible adverse events sometimes, because it's really important that before gene therapy, patients receive full checkup for the more, not only blood testing, but also check-ins with the heart system, with the liver, because with the immune response, we have to follow steroid to use steroids for a very long time. Sometimes only 1 or 2 months if everything goes well, but sometimes it's even longer. So in median, I think it's about 3-4 months. In some patients, it's even more than a year. And I think it's also should be discussed with the doctor at the time you're making a decision whether to use or not to use gene therapy. Sometimes patients saying that gene therapy is working way better than other disease-modifying treatment, which is really not having any evidence on that. At least in SMA, we have currently the data showing that all 3 treatments we have, including gene therapy, shows more or less the same results in them based on the clinical trial data. Sometimes patient thinks that I will get gene therapy and I will not need to go to any doctors because I will be out of my disease, or it will not influence anyhow my life. But it's again in SMA, even if you get gene therapy, so still you need a lot of rehabilitation process, a lot of physical therapy, a lot of the respiratory management, and sometimes nutritional management as well. So that is still important. And I already said about the long-term data, but also I think that we need to really be focused on getting full multidisciplinary care and physical therapy, because that is the key to improve the effect which gene therapy could bring to us. I think that the benefits actually of gene therapy is obvious because it's one-time solution, but still I think that we need to not underestimate the need for additional care for any patients, whether he or she was treated symptomatically or pre-symptomatically. So I will talk also about access situation, because that is a part we are not yet discussed. And I think that it's not surprisingly, but that access in gene therapy is really not equal in all the world, but also if we are mostly focused on the European community, it's also not equal in different European countries. For current state, you can also use SMA Europe Aartsma project, where we try to collect information on the current status of access to different SMA medicines. You can see map here. And we still seen, despite of having united email label, additional restriction based on age in some countries, on weight, on SMA type, on ventilation, compared to the full approved label. And I think that it's important also to know, because if in Europe it's more or less situation is I cannot say it's very good, but it's quite good. But some countries completely left behind with no access and no chance for access, and it's mostly Asian countries and African countries. And of course, one of the reasons why access and reimbursement is different in different countries is the price, of course, because it affects decision making not only of families and healthcare professionals or expectation from the treatment, but it also affects decision making of regulators and payers. And of course, we all the time asking, with such a high prices for the gene therapies, how our healthcare system can work with that. And you can see the current, like list of the most expensive treatments available, and 2 out of 6 is actually neuromuscular disease gene therapies. Why I'm talking also about access, because I think that experience learned in SMA could also help in other disease areas where gene therapies is currently under development, to not duplicate their, like wrong things or that ugly patterns, which also affect the communities. And I mean the crowdfunding, because I guess everyone heard about the intensive fundraising in many countries for gene therapy, even if there are other DNTs is available. And I think that it sometimes delay the treatment of the patient, because they hope and they wait until they collect money and refuse to get treatment with other DMTs, or just waiting for the gene therapy to be delivered just when they collect some money. But also the consequence of these crowdfunding campaigns is sometimes it's spreading, not sometimes, but most of the times it's spreading correct information, because if you tell people that you will stabilize your patient with $2 million crowdfunding campaign, you will never receive this amount of money. So sometimes it's spreading the information of miracle drugs or cure things, and again it will affect afterwards regulatory and reimbursement decisions, because the politician and decision makers is also most of the times do not have proficient medical background, and also hearing what the information is spreading around. And another thing which I think it's should be avoided in the future with gene therapies is so-called lottery program. It was run by the company as a compassionate use program, but with a blind selection of the patient allocation for treatment. So that is there was a criteria initially includes every patient in every country where there were no access in for the gene therapy, where a lot of patients just waiting in the line, and you never know when your line will be there, because each month it will be a blind selection of the few patients, and sometimes it's bring patients to the unrealistic expectations and hope, which were never happened to be true, and some mortality cases, of course. And I think that it is really important when the patient community working also with the industry to make sure that any compassionate use program on the benefit of patient was sufficient with the ethical side of the question, which was not true here. And still we are not sure that this global program was a compassionate use program, because after 3 years the company shortened the list of the countries for not where there were no access to the really treatment, but where is currently possible to find long-term solution countries, which means that only for the countries where they probably will be able to market their drugs. So that is what we can or what we have to fight. I think that in neuromuscular diseases for the future together, not duplicate these things. And what I was already mentioned that we do have in SMA a few different labels. We do have different approval labels in different countries of the globe, and it's also bring patients to to try to find any solution to receive. So, for example, we do have like what we call FDA label, because it was approved for the patients without any exclusion criteria, just based on the age of 2 years old, and EMA, which was approved the product for the patients up to 3 SMN copies, but not restricted the age of the patient, which bring the community also to discuss which label is more true, and the clinical community also to discuss the risk and benefit profile of the patients older than 2 years old and weighing more than 13.5 kg, because at the time of the EMA approval, there were no current data from the clinical trials suggested the data for the older patients. Now it exists already. The company was establishing after regulatory approval additional trial. It's now done. Everything good. The results is quite sufficient to use gene therapy also for the a bit older patients up to 21 kg. But also the EMA label bring us to the question and to restriction to use gene therapy for the patients with the 4 copies identified, for example, through newborn screening. So if in United States, patients identified through newborn screening with 4 copies can receive gene therapy. So in European countries or the countries with EMA based label will not be eligible for even eligible for gene therapy because they just have 4 copies, which is also have no clear answer why they were excluded. So I think that that bring us also to the question of design of clinical trials, which really affect then the label and then reimbursement decision as well. And additional discussion currently we have as a benefit profile for severely affected ventilatory dependent SMA population, for older population. So it's still not enough data to see whether gene therapy will provide some additional benefit for them more than other DMTs which already have this result. But there is also ongoing efforts to show this data through the data captured with a clinical, real life clinical practice through the registries. And why I'm talking a lot about the clinical trials, because from my perspective, as a patient communities, we have to work intensively with pharmaceutical companies at the very beginning of the developing stages of their products of gene therapy, and also being invited to or try to be at expert boards or advisory groups in the design of clinical trials, because what we've seen and what we learned from SMA, that data from clinical trials really affect reimbursement decision and really restrict access. That's why we still do not have access for ventilated patients in some countries, or we have some restrictions based off age or weight in other countries. And also, when we plan clinical trials, we really have to see what type of the population can benefit from gene therapy at the very beginning. So we have to include the full range of the population in such clinical trials, do not facing afterwards restrictions for them. So as an example, we do have a current trial already almost ended, and we waiting for results of IT delivery, which is another method of delivery. Then we forget, I think with Annemieke, we both forget to say that gene therapy currently is intravenous delivery. And for all the population in SMA, the company tried to develop IT delivery method for older populations, so for the patients from 5 to and more age group. And this will theoretically allow us to use lower vector to deliver the same amount of product needed for this patient. So the results from this trial we still waiting. But why I'm talking about this now, because actually while manufacturers see adult population and different subgroup of the population as a target, in the trial they or they cut the age of inclusion criteria to 18 years old, which we again see as a possible possible problem for the reimbursement for the adults, for example, in the future, because really that is a lot of their unmet needs in adult population as well could be addressed with gene therapy as well. And of course, I think that it's important also to mention that not always in the clinical trials we get all the answers about the safety, and some bad news comes also from the clinical practice. So, for example, the cases of acute liver failure and death cases, and thrombocytic microangiopathy cases, which were only this data about these severe adverse events were only established after gene therapy were approved, and never happened in clinical trials, but were happening and continue happening in the real world usage of gene therapy, which is of course require additional concerns of the patients. And I think that I will try to conclude because of the time. Sorry for taking too much time for this. I think that when we use gene therapy, we have to have informed decision, and to have in mind always that early treatment is the most important, and which make us probably to consider which or what type of of therapy, any therapy, will be available to the patient faster. Because if you talk about the gene therapy, sometimes giving gene therapy takes time, not just because it's too hard, it's not, but because gene therapy product, at least in SMA, is really specific to weight with a very, and the production is basically done for each patient, depending on the weight of the patient. So it takes some time, and sometimes it's like few weeks, sometimes it's few months for logistic things. And in this period of time, it's really important in terms of SMA to start another DMTs before gene therapy just to not lose motor neurons, just to keep patient not affected with any additional symptoms of motor neuron loss. Also, I think it's when we discuss we have to talk about the expectation management. And of course, there is a huge need of psychological support. But when we talk about the pre and after dosage monitoring, we have to be sure that additional tests could be required, not only the blood testing, but doctors in the centers can require some more testing and some more days of hospitalization before dosage. And also you have to think that after dosage you also have to be for some time in the clinic and under the close monitoring of the doctors. Patients should be informed of the early signs of possible adverse events and how to deal with them. In what cases you really need to go faster to the clinic because some symptoms can be really dangerous in the future and can be the sign of really severe adverse events. So parents should be trained and informed where to refer it. And sometimes also the challenge here is exchange between healthcare professional teams, because generally there is not that much gene therapy center, and it could be the situation when gene therapy is in one city and the patient living and being under the team in other hospital which is not providing gene therapy. So here is also importance of communication between the doctors. And what about the long-term follow-up? I would only just saying it's not only about the safety monitoring or managing steroid dosage, not only the motor scale measurements to access the efficacy things, but it's again, it's about physical therapy and multidisciplinary care as the main part of working with the patient to gain more efficacy and more results. And again, I think that compliance of the family and collaborative efforts of the family with their doctors is really important, but because sometimes we facing the situation that patients receiving gene therapy just withdraw from any monitoring of the doctors, thinking that they don't need it anymore, but that it's really not what should be happening. So to conclude, I will just give some maybe takeaway messages for other patient groups, non-SMA patient groups, just what to do if you have gene therapy in the market, just not to duplicate the struggles we try to manage currently in SMA, and to learn from our experience. I think that is the most important thing is really proper communication and correct information about gene therapy in your community. It will help to manage expectation, because gene therapy is really not a cure, and what to expect we really have to manage within ourselves as well. And I think we have to really try to deliver to our communities that gene therapies really have some limitations, both in efficacy and also some safety consideration. And the fact that gene therapy is really still new, even after 5 years of SMA gene therapy, it's still new for us. And there is really need for proper training, education, and information, and for patients, for healthcare professionals, and also for society. And I think that, really not, my message will be not to duplicate some sad experiences about the expectation or some false messages spreading around gene therapies which we had in SMA in other disease area. Thank you. I will try to get rid of my slides now. And we are ready, I guess, for the questions.
Thank you so much, Olga and Annemieke, for those very interesting presentations. So the first question, I think maybe we'll do one or two only, because the time is short. So now that have I pronounced it right, Zolgensma has been trialed in all the patients with a fixed dose, if it gets approved for all ages, how effective would it be for adults? Could it be more or less effective than Spinraza or Evrysdi?
I will try to answer that, and Annemieke will help me if I mistaken something. So I think that also we do not have any trial which compare the effectiveness of any DMTs for now. So we really cannot talk about whether gene therapy could be more effective than Spinraza or Evrysdi. Because there is no data which we can use, and we cannot just make a direct comparison because the trial population is really significantly different from each other. So I think that we will not really find an answer for this question. And if we talk about their approval for all ages, which we really hope for, it's really hard to explain or answer by now because we haven't seen yet the data and we only expect it, I guess, next year. So at least that is a hope. And theoretically, if we using intrathecal injection, and here probably Annemieke, I will need your expertise. So if we use less viral vector and we deliver it not to IV but intrathecally, so we deliver it to the target part of the body. So then we can use really small amount of adeno-associated virus, which bring us to the low dosage, but also lower dependency of the dosage of the active substance inside. So whether I am right or not.
Yeah. So what you will get is locally you would get a higher dose because you inject intrathecally, so you're close to where you need to be, the motor neuron. So locally the dose will be higher, but systemically, so the rest of the body, the parts that you don't want to target, especially the liver, those doses will be lower because, well, the total dose for the whole body is less. So that is indeed the case. But of course, the question is, will this work? Yes or no? I think that is that's the point that we're now trying to figure out. And also, so will this work and will this be safe? Because locally the dose will be higher than normal. So will the central nervous system tolerate that? And will that be effective? So will it actually work if we do it like this? So yeah, we like Olga said, we have to wait and see. I'm not going to speculate about ongoing trials.
Yeah, and I think that the question, part of the question was also because the current trial was only recruiting the patients up to 18 years old, but not any adults. But still, we will have data on like teenagers, 16, 17, 18 years old. So probably that is not much different from adults. I think in terms of, of the growth of the body. But for me, the question sounds like whether it will be also sufficient for the regulators. Even we in the patient community have this question whether it will be working in adults, while we do not and we will not have a data, whether the regulators really will approve for adults based on their data only received under 18 years.
And of course, one adult is not the other adult. I mean, there's differences. There's differences between some 20-year-olds. Some will be much better than others, and there's a difference between a 20-year-old and a 40-year-old as well. So, yeah, that's again. I'm also not going to speculate on what the regulators will say, but this is really something that I think we need to wait and see. But regardless, what this will do, it will slow down progression of the disease. So it's not realistic. If an adult is in a wheelchair, it's not realistic to think that they will stand up and be normal. I think that is, and I think that message has been clear, but I want to stress it again. It's good to have the right expectations of what this can do.
Thank you for your answers. And the last question is, what is a placebo in gene therapy? An empty vector, physiologic solution injection?
Yeah, so this is it's an interesting question, actually. So it is just saline. So yeah, the 0.9% salt that our blood has as well. It's not an empty vector, and the reason is if you would deliver an empty vector, you would get an immune response to that, and then later you would not be able to treat your placebo group. So usually what happens in these trials is that the placebo patients get treated later. So everyone gets treatment at time point 0, half as placebo, or 1/3 gets placebo. And then after 1 year or after 2 years, those that had received placebo now get the gene therapy. If you give the empty vector, you cannot give the gene therapy later because these patients have now been immunized to AAV. So that's why that is not done. It's they get saline, which also means that with the side effects that gene therapy can achieve, it's not really that much a double-blinded trial, because you will generally not get super nauseous from saline injections. Well, very often from high-dose AAV, you will get super nauseous. So that's difficult to really remain blinded then.
Okay, thank you so much. Thank you, Olga. Thank you, Annemieke, for this talk.
Thank you.
And thank you, everyone, for listening to it. Have a nice day.
Thank you. Have a nice day.
Bye.
Bye.